Good morning, good afternoon, and good evening all. Thank you for taking time from your busy schedule for joining us today. We appreciate all of our attendees from the West Coast, East Coast, and a significant number from Europe. My name is Sharif Hanala. I am the product manager here with the Bioprocess Chromatography Group at Bio-Rad, and I will be the moderator for today's session. Now, we hold these webinars every so often when we feel that we have some significant insight that we can share with the industry to help you move your work forward and as we work together to tackle the industry's biggest challenges. Today's webinar is entitled Using Mixed Mode Selectivity to Target Purification Challenges or to Address Purification Challenges. Today is a part of a webinar series that I will speak about our second webinar coming up later this month at the end. But first, I'd like to go through some logistics. Today's webinar will last approximately an hour. The presentation will run between 35 and 40 minutes, and then we hope to have about 20 minutes worth of discussion and questions. We want to see this very much as a conversation, not a one-way communication, so please post your questions. You will notice that there's a Q&A box on the left-hand side of your screen. If you enter your questions in there, I will keep track of them, and if there are urgent ones, I will pass them on to the speaker. If not, we will take ample time at the end to address your questions, or perhaps follow up later with more data and conversations after the webinar. Any technical difficulties, please enter your questions also in the same Q&A box, and the good folks at On24 will help us with those. It is now my distinct pleasure to introduce our speaker today, Dr. Xumei He, a very valuable colleague here. Dr. He has been with Bio-Rad for a significant number of years. She has earned her PhD in biological chemistry from the University of Minnesota. She went on to take on technical roles of increasing responsibility in the industry, but for the past few years, she has been working here with her team very closely with customers to help redesign purification processes as well as address purification challenges from anywhere in the world. So without further ado, I'd like to hand the floor over to Dr. He so she may begin her presentation. Xumei.
Thank you. So hello everyone, and today in my talk I will focus on two mixed mode chromatography media. You see on the next slide. So the first one is nucleus C prime, and the second one is ceramic hydroxyapatite, also known as CHT. I will show you the structures of the chromatography resins and the possible interactions between the media and the biomolecules. So with the knowledge of all these interactions, I will show you how we can develop an effective purification method and how we can tackle the purification challenges we have, because we know protein purification is not always straightforward. There is always problem unique to a particular molecule or a particular purification process. So here you show the chemical structure of the ligand on nucleus C prime, a hydrophobic cation exchange media. So on the ligand, there is a carboxylate with a pKa at around 5, and for the benzene ring, so on the other side of the ligand, you see there is a benzene ring, and sandwiched in between there is a amide bond. So on top of the chemical structure, you also see a model that shows the distribution of charge and hydrophobicity on this ligand based on calculation studies. So the red color shows you the hydrophobic benzene ring, and the blue color shows you the charged carboxylate. Because of this carboxylate group, the charge state of this ligand is pH dependent. If we do purification at higher than pH 5, this ligand will carry a negative charge, and nucleus C prime will act like a weak cation exchanger. At pH below 5, this ligand is more like a hydrophobic interaction chromatography media. With the combination of all the three structural elements offered by nucleus C prime, there is a unique selectivity we observe, because now we are using three criteria to distinguish target molecules from impurities. However, the hydrophobicity of nucleus C prime offers the more salt tolerant and traditional ion exchange resins. Because the ligand is linked to the media through the benzene site, you can tell it's not totally exposed to interaction with protein molecules. So we often see the hydrophobicity of nucleic He is not as strong as conventional hydrophobic interaction chromatography media. The presence of the amide bond also creates a network of water molecules on the surface of the resin. This helps prevent hydrophobic interaction not as strong as the HIC, and also it prevents the dehydration and denaturation of target molecules, a problem we often encounter in hydrophobic interaction chromatography. In general, the purification can be conducted under milder conditions with nucleic He as compared to the conventional HIC resins. So this is just a graphic way to show you the possible interactions between nucleic He and protein molecules. And such interactions can be modulated through the change of buffer pH and conductivity, just like any other ion exchanger and hydrophobic interaction resins that you are familiar with. Next, I'm going to talk a little bit about ceramic hydroxyapatite. The ceramic form of hydroxyapatite is much stronger than the naturally occurring hydroxyapatite. It also offers multiple interaction functionalities for protein purification. First, let's look at the left-hand side. We see the phosphate groups on CH. They interact with the amino acid residues on protein, like the amine groups, and this kind of interaction is sensitive to buffer pH, sodium chloride concentration. There's also calcium site on CH. They form complex with the carboxylates on protein molecules, and such interaction can be modulated by buffer pH, phosphate concentration. And DNA is one of the main impurities we want to remove from purification, because DNA, a lot of time when we open the cells, when we harvest the culture, the dead cells release DNA molecules. And since DNA molecules have a lot of phosphoryl groups, they form very strong complex with the calcium sites on CH. So we can control the buffer pH and salt concentration, phosphate concentration, to achieve selective binding and elution of target molecules while retaining DNA on CH until we strip the column. So now I want to move on to method development, because when we first use mixed mode media, we are sort of nervous because we don't really know what kind of interactions will take place between the target molecule and the chromatography media. Because when we deal with a new protein molecule, the only information we know is from the sequence about the PI, the possible PI of this protein, and the molecular weight. Besides that, we don't really know if there's any net charge on the protein, what kind of hydrophobic residues on the surface that we can use for interaction with chromatography resins. Here, we want to suggest a more generic approach. That's a simple DOE screening. We want to use conditions shown in this table to cover pH from 4 to 8 and sodium chloride concentration for the purification of lysozyme from E. coli extract as an example. The reason why we choose pH 4 to 8 because it's very commonly used in protein purification, gentle to target molecules. And then for binding, we choose the range from 10 to 400 millimolar sodium chloride because this is also the most common concentration range when we harvest from cells. Then for elution, we look for a wider range of sodium chloride concentration. That's because we don't really know what exactly is the interaction on the column, so it could be ion exchange type or it could be hydrophobic interaction. So we want to see from low concentration 10 millimolar up to 1,000 millimolar of sodium chloride to see how we can achieve elution. However, in reality, you may have a protein different than lysozyme. So you may know more about the stability of your protein, then you can change the range of the pH and sodium chloride concentration based on your knowledge about the stability. And then to look at the effectiveness of the purification, we check the yield and the purity of the target protein, in this case lysozyme, from the column. So we want to see, after the selective binding and selective elution, what kind of lysozyme purity and yield we can achieve as a way to evaluate the effectiveness of the purification. This can be done in a spin column format, and totally there are only 11 experiments. You can pack a spin column with about 50 to 100 microliter of resin, and we include 3 standard points as a way to assess the reproducibility of the experiment. Now let's see the results from the DOE screening. This can be done really quick because the DOE, the binding, the elution can be done within 30 minutes, and we do gel analysis to check the purity within an hour. Then we can plug in the binding capacity and do a DOE analysis to get a roughly purification condition or the sweet spots for the purification. As you can see on the slide, the binding condition identified by this DOE exercise is low pH at pH 4 to 5. And with modest concentration of sodium chloride, 10 to 150 millimolar, we can effectively capture the target molecule from the cell culture harvest. And then for elution, it seems that elution of lysozyme is not sensitive to buffer pH. Pretty much from 4 to 8, you can achieve elution as long as the salt concentration is higher than 800 millimolar. And now we can pack a column to confirm this result. So on the right-hand side, I showed you the condition for column chromatography, and we choose 2 conditions for binding. One is at pH 4, the other one is at pH 4.5, because we want to check the robustness of this condition. And now you see the table shows you the dynamic binding capacity of the target protein, the recovery, and the purity after the separation. They are very consistent. And here is the purity check of the purified lysozyme. Although the binding was done at pH 4 and pH 5, we didn't really see any significant difference in product purity. So it's single band purification that you often see only when you do affinity purification. So the purification is really great by a simple DOE screening and then scale up on the column. So does this only work for lysozyme? So we have this question, and we look further with a couple other proteins. So if you can see the table clearly, you see the PIs of this model proteins range from around 5 to 9. And the molecular weights are quite different. Besides this, we don't really know what kind of charge residues on the surface, but we use this protein models to check DOE, if DOE is an effective way for us to come up with a purification method. So on the right-hand side of the table, you can see the sweet spots we identify from DOE. So for the first 3 columns of the proteins, the BSA, the carbonate anhydrase, and lysozyme, if you take a closer look at the binding condition and the elution conditions, you may notice that these proteins interact with Nilvia C' like cation exchange resin, namely binding at low pH, low salt, and elution at high pH, high salt. So the second category of protein is corn albumin. The binding, again, takes place at low pH, low salt. However, the best elution condition is not at high salt. It takes place at pH 6 with just 500 millimolar of sodium chloride. So this shows us the interaction mode of coalbumin is different than the top 3 proteins. And then for the last 2, we know that we need to add some salt, sodium chloride, in the buffer in order to promote hydrophobic interaction to achieve a better binding capacity. So again, before we do this DOE, we have no knowledge about interactions, but after we finish the DOE and do data analysis, now we have a better picture about possible interactions between target proteins and the chromatography resin, and then we can fine-tune the purification based on these results. Now, if you are a more conventional chromatographer and you prefer running gradients to study interaction between your target molecule and the chromatography resin, you can do so with Nubia C'2. So first, in order to have the protein bound onto Nubia C', we found that you can begin with pH 5 with whatever sodium chloride concentration already in your sample. And then you run a pH gradient. With the increase of pH, we can create charge repulsion between the ligands because we know the carboxylate will become negatively charged and your protein molecules will lose some of the protons during this pH gradient. And let's see the effect of pH. We can determine what kind of interactions are involved. And then we can also look at the effect of sodium chloride concentration. By increase or decrease the concentration, form a gradient on the column. And sometimes the purification could be more challenging, and you cannot just by changing pH and sodium chloride concentration in your buffer to achieve the separation you want. At this time, we may need to add additives, which I will give you more examples later. But anyway, once you get enough knowledge, it's very easy to convert a gradient purification into a step elution. So now I want to give you an example why we have to look into both the effect of pH and salt concentration. In this case, the target molecule is a glycosylated enzyme with a pI at around 5. And we found that this enzyme can be bound effectively at pH 5 with low concentration of sodium chloride. Now I also show you the structure again of the ligand, the pKa at around pH 7 at 4.7. So at pH 5, the ligand likely carries half of the ligand has negative charge, and the other half of the ligand is non-charged. So we think in this case the binding is through hydrophobic interaction as well as cation exchange. Now on the right-hand side, we see if we apply a pH gradient on the column, the target molecule is eluded. However, you also see a lot of impurities on the SDS page gel because we think when we raise the pH, we change the charge state of both the protein, target protein, impurities, and the ligand. On the other hand, if we keep the pH constant on the left-hand side at pH around 5, but we raise the concentration of sodium chloride in buffer, in this case, you see we selectively eluded just the target molecule, giving you a much better separation. So it's important to look into both effects in order to achieve a satisfactory separation of your target molecule from the impurities. So now we come to real cases in purification. What kind of challenges we have? First, we need to deal with impurities coming from the host cells, such as the host cell proteins and DNAs. So the first case I'm going to talk about is the purification of an IgM molecule. As we all know, IgM molecules are much larger than IgGs. often, these days, the expression level of IgM molecules is not as high as the monoclonal IgG molecules. We can really get 6 to 7 grams per liter kind of expression. And a lot of time, IgM molecules are found forming complex with DNA. They are not as stable as IgGs, so they're very sensitive to buffer salt and pH. On the other hand, they're highly glycosylated, making the charged residues not accessible for interaction with chromatography resins. And we can really use protein A because IgM molecules are sensitive to low pH, and there is no other form of affinity purification currently for IgM molecules. So this IgM molecules we show here is from a client, and it has all the problems I mentioned earlier. The goal for this new purification workflow is like we want to have a milder condition to protect the activity of this IgM. At the same time, we want to have minimum feed stream manipulation so we can cut the purification process shorter and do it faster. At the end, we want to have the target molecule active and stay aggregate free. So this is done by a 3-column workflow. The first column is AG1XA. We use that for removing DNA and other DNA binding molecules such as histones. And then we use NuVH-C prime to remove the bulk of impurities. You will see that on the gel later. And at the end, we use ceramic hydroxyapatite as the final polishing step to further take care of DNA contaminants. So the main contaminant in this IgM expression system is the serum albumin, which has a pI of 5, very close to the target molecule's pI. And we've tried to use anion exchange chromatography resin. We found that both the target molecule and the impurity serum albumin are bound, and they are not well separated. And this molecule, again, is not stable under low pH and low salt concentration. It precipitates if we try to dilute the feed to load it onto a cation exchange column. It forms aggregates on HIC column and has a poor binding on the protein A column. Then how do we do the purification? We found that actually it's very interesting. NuVH-C prime has a very good selectivity of this IgM molecule. We did a quick DOE screening again, and we found the most important parameter in this purification is the pH of your feed stream. If we control the pH to pH 6.5 and above, NuVH-C prime can bind the target molecule selectively, or the serum albumin will appear in the flow-through. And in this case, the binding is not sensitive to the sodium chloride concentration in the feed stream. There is no need for dilution, so we can increase the yield because we are not denaturing the IgM molecule, and also we can reduce the cost for sample manipulation. And the elution of the target IgM molecule can be achieved at 400 millimolar sodium chloride at pH 7. And the gel show you after the C prime column, we can remove the serum albumin and keeping the heavy chain, light chain you can see on the gel, and a little other impurities. And then we move on the final polishing step on ceramic hydroxyapatite. So the left-hand side of the slide shows you the eluate from NuVH-C prime. It contains 400 millimolar of sodium chloride. We load this onto the CHT column directly. With 400 millimolar sodium chloride, all the whole cell proteins appear in the flow-through fractions. And then we raise the buffer phosphate concentration to elute the target molecule. Here is the purity analysis. On the left-hand side, this is the HPLC SEC analysis. Show you that the aggregate content in the final preparation is less than 0.6%. You can compare this from the material purified from the previous. It doesn't show up on the screen. The HPLC color is too light, I'm sorry. So for the previous protocol, there is contamination from aggregate, also from the degradation of the target molecule. With the new workflow, you clearly see there's only monomer eluted from the hydroxy apatite. We also check the OD 260 to 80 ratio for the feed and for the final material. This is a simple method to assess the DNA contamination level in your preparation, and we increase the ratio to 80 versus 260 from 0.9 to 1.8. So this value is very close from the calculation based on sequence of this molecule. And now I want to switch gear to another important class of molecules. That's the recombinant viral antigens. This recombinant viral antigen also is heavily glycosylated. It's not really bound by cation exchanger, anion exchanger, and we've also tried glycan affinity column, or gave very poor selectivity and poor binding capacity. However, if we reduce the pH of the culture harvest to pH 5, we found the target molecule is bound. And then we try to wash out the impurities. As you can see here, we use a gradient from pH 5 to 8 to wash out impurities, keeping the salt concentration at a relatively high level, 250 millimolar of phosphate and then 250 millimolar of citrate. So for the first gradient, you don't really see a lot of protein bands on the SDS page, lane 2, 3, 4, and 5. However, by OD, we know the DNAs are washed out from the column. And then we hold the pH at 8, keeping the salt concentration high. For lane 6, you see more protein impurities are washed out. So when we are ready, we start another gradient to elute the target molecule. So for the gradient 2, we keep the pH at 8, but we reduce the salt concentration. And you see only the target molecule was eluted. And based on this result, we know that the impurities are bound by nuclear C prime through charge interaction, and the target molecule actually is bound through hydrophobic interaction because it was eluded by salt concentration reduction. So this is very interesting. It's like we developed the method, but we also know more about the interaction, and we can fine-tune this purification based on our knowledge during protein purification method development. Now, I want to talk a little bit about product-related impurity removal. Namely, they are the fragments from degradation of the target and also from the aggregation of target molecules. So the first case is about the removal of a monoclonal antibody fragment on nucleus C prime. We found that most important parameter is the binding pH. Now, again, you see this pH 5. It seems to be a magic number. So at pH 5 with 100 millimolar, 25 millimolar sodium chloride, we can have the fragment in the flow-through fractions and then keep the monomer on the column until we raise the buffer pH to 6.2. You see in the middle there is a very sharp elution peak. And how do we confirm the removal of the fragment? We ran STS page with the fractions collecting from flow-through and excised this band from the gel and ran mass spec analysis, confirmed this is a light chain fragment. The HPLC On the right-hand side, also shown the orange dashed line is the load, and the green line is the LUH from new VSC prime. You also see the clearance of the fragments, the little bump at the bottom. So for aggregate removal, it's also quite straightforward. In this case, the second map, we load the material at a low pH with modest concentration of sodium chloride. This is based on DOE screening again. And then we raise the pH and salt concentration in buffer to elute the target molecule in a monomer form. We collect fractions for HPLC analysis. As you can see on the next slide, the load contains about 10% or 11.5% exactly of aggregate. And after we finish the purification on new VSC prime, we see that the LUH we collected during the gradient contains only 0.2% of aggregate. And then the strip portion just contain all the aggregates we want to remove. So you can see how effective for aggregate removal. However, things are not always this simple. Sometimes we do have to use additives to enhance the resolution between the target molecule and unwanted species. So this table gives you a list of all the common additives we may use for separation on new VSC prime. As you can see here, depends on the structure of these molecules. They may change the interaction between the target molecule, impurity molecules, with the chromatography media, through changing the electrostatic interaction, hydrophobic interaction, or the strength of the hydrogen bonding. We cannot really predict, and we have to do experiments. In the next case, I will show you how we do that. So the goal for this separation, again, is for removing aggregate from a monoclonal antibody preparation. So the goal here is like we want to remove the aggregates. At the same time, we want to maintain a high recovery of the target molecule. So on the figure, you see if the white area is bigger, we say the recovery is better. And then for aggregate removal, the starting material contains 96% of a monomer. The rest are aggregates. And then our goal is to achieve close to 100% purity of monomer. So this result is very interesting because we take another mixed mode media. It's also a hydrophobic cation exchange media from the market and compared it with a new VSC prime. As you can see here, some additives like urea, the propylene glycol, glycine, didn't really work for aggregate removal. On the other hand, arginine works better. However, we can see a trend here. It's like elution of the target molecule from new VSC prime can take place at a lower pH compared to media A. This is a benefit in general because we know at neutral and alkaline pH, the monoclonal antibodies tend to undergo deamidation and hydrolysis. So if you can do the purification at mild acidic pH, that's a benefit. And in this experiment, we also want to see, we're curious, how different is new VSC prime than a traditional hydrophobic interaction chromatography. So you see here, like the target molecule can be eluted at 0.6 molar ammonium sulfate. We all know that ammonium sulfate is used to promote hydrophobic interaction. However, the target molecule can be eluted with a high concentration of ammonium sulfate. So we think the hydrophobic interaction, again, is not very strong on new VSC prime, which is a lot of times better than a traditional HIC purification. On the other hand, we do see the different effects of additives. And again, arginine works the best, so we decided to do further investigation. So let's see arginine. we see the pH has an effect, and the concentration of arginine also change the recovery and aggregate removal. So we want to pack some columns to validate data from this plate screening. There were 3 conditions we chose from the plate screening. The first one is on the upper left-hand side. This condition, there's no additive or arginine added to the buffer, but the elution was done at higher pH. And then we picked 2 spots on the right-hand side, lower right-hand corner. One has lower concentration of arginine. We want to see at lower pH how the concentration of arginine affects the separation effectiveness. And the results are shown here. So for the first condition at high pH without arginine, we see a good improvement of monomer content. It improves from 96% to 98.4%. However, the recovery of the target is modest, not the best. And then we also checked the second condition is at low pH and low arginine concentration. This condition gave the best aggregate removal. However, we also lost a lot of target molecules. So for the third condition, it has a good balance of both recovery and purity improvement. When the pH was low with high concentration of arginine. So the key here is we need to know the spec for this purification and then decide what is the best condition for the purification. In this case, it's about the FC fusion aggregate removal. We all know that FC fusion usually is not stable and tend to aggregate. For this aggregate removal case, we found that guanidine hydrochloride worked the best, and also the separation is pH dependent. It didn't work at pH 7, but at pH 5, we were able to get a condition with about 0.5 molar of guanidine hydrochloride. We can retain the aggregate on the column and recover most of the monomers. Now, besides taking care of the impurities, we also want to have a good recovery of the target protein and maintain its activity. So here the example I want to show you is the purification of a recombinant protein from E. coli extract. Again, we compare 2 mixed mode media. One is Nuvia C', another one is also a hydrophobic cation exchange resin. So we found that if you see the green color of the column, that means the binding is good. There is no target molecule in the flow through. So for media A, you see no matter you are using a high concentration of salt with high conductivity, or you choose the pH from 3 to 8, you always have a good binding. On the other hand, for Nuvia C', the binding takes place between pH 5 and pH 8 with no salt to up to about 200 millimolar of sodium chloride. Well, when we first glanced through this data, we thought, okay, Nuvia C' doesn't really work if we have extreme pH. But in reality, most of our culture harvest has a pH from 5 to 8, and then with modest concentration of sodium chloride concentration. So we know based on this data, both resins are suitable for capturing of this target molecule from the harvest. And then you have a question when you look at this graph. Like the binding of target protein on media A is not sensitive to pH, it's not sensitive to salt concentration, then how can we do elution? Your concern is confirmed on the next slide. You can see elution of the target molecule from media A is only possible if you use high pH, like up to pH 9, or you use a lot of additives from 30% to 45%, or you use high concentration, 2 molar of sodium chloride in order to elute the target molecule. On the other hand, elution from nucleus G' is a lot easier. Depends on your next purification step, whether it's a hydrophobic interaction chromatography or a ion exchanger. You can choose to elute the target molecule at high salt concentration or low salt concentration. You can also choose your elution pH, and the elution can take place without adding any additives. So this is a very, really a benefit if you try to fit this chromatography step in the process. It gives you more options and makes the manipulation minimum between columns. A lot of time when we screen for a different chromatography media, the first thing we will do will be determining the dynamic binding capacity. However, purification is not just about binding, it's about elution and recovery. So my suggestion is when you are comparing different chromatography media, you should always look beyond binding. Now, I want to talk a little bit about process economies. And I've already given you example, like the viral antigen case and the IGM purification. We only need to adjust the pH of the feed a little bit in order to have an effective binding, and then we do selective elution. You don't really have to manipulate the feed a lot. And for the recombinant protein purification case, nucleus G' offer you a lot of choices in terms of elution, allow you to move on to the next purification step without manipulation of the feed. This saves your time and your cost on labor and material and facilities. So here is a brief summary of my talk. I've shown you the new and unique selectivity for mixed mode chromatography. And then this kind of selectivity can further enhance by the use of additives and by the variation of buffer salt concentration and pH. And also for mixed mode chromatography, we often see they all have good salt tolerance and wide pH window for separation, which will allow you to do easy step transition between chromatography steps and other purification steps. And you can also choose a condition that's gentle for protecting the activity of your target protein. The method development is quite intuitive. All guidelines you learn for the ion exchange chromatography, hydrophobic interaction chromatography, chelation chemistry still apply. So we can gain knowledge during method development, and we can refine the purification condition based on our knowledge from this exercise. And I hope with all this information I give you today, you get a better picture about mixed mode chromatography. And if you need further information, you can always come to our website to search our mixed mode products. And if you have technical questions, you're welcome to contact me directly with my email here. Thank you for coming. And I'll be ready to answer your questions.
Excellent. Thank you very much, Xumei, for another information-filled presentation. I hope that the concepts and examples that were shown today have sparked some imagination in the minds of the audience so they could put some of these concepts and ideas into good use in their day-to-day challenges. I hope that it was valuable for everybody. Now we are ready for some questions. I have several questions from the audience. I will read them in order, and we will answer as many as we can. Also remember, please, that if you have any further questions or request further data behind some of the applications we presented today, we'd be happy to provide those after the webinar. One of the questions that I got is actually from more than one person is, when do you consider using an additive when you're using mixed mode chromatography?
So as the examples I have shown you, firstly, I usually just look into the buffer pH and salt concentration. Only if these two conditions are not helping, we will try to add in additives. And the additives I usually recommend is arginine because it has been used widely in industry and in approved purification process. So this, a lot of time it works. And also, when you handle arginine, it's not as messy as like urea, for example, or guanidine hydrochloride.
Mm-hmm. And in your experience with the resin, what percent of the time do you think we would have to resort to, like in the case of C prime and some others you use? Do you notice a difference between the number of times you have to use an additive to get sort of proper elution?
Well, I would say from my own experience, it's about 10%. So for the majority of the cases, you don't have to, even for IGM molecule, the aggregates were removed without using any additives.
Okay, cool. Well, the next question is also related. So the next question was, how do you select the additive? So I think you may have already answered a little bit.
Yeah, I would always use arginine first.
So start with arginine. That's a pretty good rule of thumb. Also, the table that's available in the presentation is also a pretty good guide to help people understand what interactions we're modifying with the addition of the various additives.
Yes.
Okay, I also have one question from the audience on slide 18. I'm not sure if we have the data to perhaps answer this directly, but it was asked that there was some amount of tetraborate used in the elution for the viral antigen. Do you have an idea why that was added to the elution buffer where we decided to elute the actual target molecule after the washing step?
Well, let me talk a little bit about the history. This was done in collaboration with a client. And when we first suggest they should raise the elution, they should do the elution at pH 8, that was the only chemical available in their lab for controlling the buffer pH. And since then, they have already switched. They are no longer using the tetraborate. In other words, you don't have to use these chemicals if you don't like it.
Okay.
It's just for pH control purpose.
Just for pH control. Okay, that's a good answer. I'm getting a lot of questions here, so I'm going to go through them as fast as I can. One of them is pH 4.5 to 5 recommended for binding when you're considering electrostatic properties?
Oh, actually, my answer would be pH 5 or 4.5 is more for promoting the hydrophobic interaction, because at this pH, the ligand doesn't really carry that much of charge interaction. Although we know pKa is about equilibrium, you always have a portion of the ligands are negatively charged, but the majority at this pH will be neutral. So it's a combination, like one of the cases I show you, it's a combination of hydrophobic interaction as well as weak cation exchange interaction.
I see, because now we're operating around the pKa, so you're basically operating in the hydrophobic domain.
Yes.
And as you go up from there, you diverge from there, then you're flipping the charge on that.
The ligand.
On the ligand to cause that repulsion. I hope we've answered that question. There's one question here that I will answer at the end, but I do have another technical question. A person here says that they've used hydrophobic mixed modes before, or other mixed modes. They're not really sure how to quantify the structure. But they've noticed quite a bit of tailing when they're trying to elute these from these resins. So they're just wondering if you have any comments on how to reduce the tailing, or if you could explain it.
First, I want to emphasize, like for the base speeds we use for making this new VSC prime, we've already optimized the structure, the particle size, and the surface properties. So we don't really often see tailing of elution. The eluate can always be collected within 3 to 5 column volumes. That's our experience. And you've also seen on my slides, the elution peak is always sharp.
Right.
On the other hand, if you do see tailing of your elution peak, that indicates that there is really mixed modes involved in the interaction between your target and the chromatography media. So if this happens, we want to look into your condition and want to look into your method development history to tell what kind of interaction is the main force causing the binding of the target molecule on the resin. And we can start from that, help you improve the elution efficiency.
Okay, I understand. So perhaps there may be a way to reduce the salt, pop the pH to create a more sort of drastic set of conditions where the target protein could just come right off.
So we need to identify the main interaction.
The main interaction, okay.
And disrupt that.
I see. Okay, it may have something also to do, I guess, with the base matrix.
Yes.
that the ligand is on. Although this is pseudo-affinity, the base matrix does play a role in as we develop resin products, we've noticed that different matrices still have an impact on tailing, irrespective of whether developing affinity or sort of pseudo-affinity.
Yeah, it could be due to the mass transfer efficiency for the molecules to get in and out of the matrices.
Right, I see. Okay, well, I think that's a good answer. Of course, we'd love to look at your conditions and help you with that. I mean, this is what we're here for. This webinar is really an invitation for you to interact with us. So, we'd be happy to look at your conditions, of course, under confidentiality. One of the questions here I'll answer myself. It's as a relatively new resin, Nuvia C prime. To our knowledge, do we know that it is used in GMP manufacturing? I can comment on that. The resin is relatively new in this industry. I would say about 2 years out, and it is being used in a variety of preclinical programs, as well as I would say less than 10 clinical programs, early stage. But the robustness sort of and the utility of the resin has certainly demonstrated itself. And as a class, mixed mode resins are becoming obviously a lot more popular because a lot of the molecules that are coming into the pipeline from upstream are posing these challenges that call for that. So there is really no need to worry about mixed mode resins sort of being around or being successful for a lifetime of a product into commercial scale manufacturing. So I think these are all the questions that I have right now. If there are any more, I mean, we still have 8 minutes or at least 6 minutes. So I encourage you to type your questions if you have any. Just type them right into the window, and we would be happy to answer them. And if there's anything else that you'd like to comment about on today's webinar, please go ahead and enter it, and we'd be happy to share it with the audience. Do you have questions? Okay, so the presenter has no questions, and I certainly have no questions, and I hope that this webinar was as valuable as we hoped it would be. I'd like to remind everyone that this is a first of a series of webinars, and as I said, we can do a lot of these, but we prefer to do less of them and wait till we have some real insight that we can bring to you. It just so happens that this month we have 2 webinars. The next one is a little bit of a higher level presentation. It will deal mostly about process efficiency and ways to compress processes, perhaps by changing the order of chromatography unit operations or using some of these unique selectivities to drastically reduce dilution or things of that nature. So if you're operationally minded or part of your task to look at sort of whole process, I invite you to join the webinar next week. We'll have a different presenter, and there will be an email reminder going out to the group, so please make sure to sign up for that on the 24th, I believe. I got one more question, so I'll ask that again. I got 2 more. So another question about the slide deck, if the slide deck will be available. The slide deck will be available if you write to us. We are not going to put it on the website right away. We have a variety of other slides also that we can share with you, so we really encourage you to reach out to Xumei or myself or your Bio-Rad representative and ask for the deck, and we would be happy to send it to you and answer any questions that you may have. I also have another, do you have mixed mode phases for small biomolecules? I'm not sure what they mean by small biomolecules. I assume that these are sort of I mean, it'd be nice to know what range of molecular weight you're talking about. But as long as you have a molecule that has some proteins in it, right? So some range of amino acids, you could think of these as potentially HCPs. These are also small biomolecules. So you can get a resin like this to operate on anything really. We've demonstrated an example where they have an IGM where you have like a pentamer all the way down to some very small HCPs. We haven't shown that or lysozyme. So yes, we do. So I would not really segregate in my mind small biomolecules versus large ones. We think of these more as sort of therapeutic types of molecules. Antibodies being large, but then we have also viral antigens and enzymes and fragments and all of these things. As long as they have some functionality on them
for interaction.
For interaction, we can basically manipulate that and a purification. Hopefully, we can do it well.
So if you have
under 1,000 Dalton is the
peptides.
Peptides, right? Okay. Thank you for putting that in.
If you are interested in testing, we can send you samples.
Oh, we have something we can test. Okay. So yeah, perhaps if this doesn't work, we are constantly trying to find resins that would be of value to you. So that may be something we can certainly do. At this moment, I don't think we have something specific for this size range. Is that a fair answer?
I think the size is not really an issue, just like you answered earlier.
Yeah.
As long as you have amino acid residues with different properties, we can always explore to play around with the interactions.
Okay. So yeah, I would say let's try it.
Yes.
Right, 1,000 and below.
Yeah, and at the same time, we are actually exploring different ligand structures. Some are more hydrophobic, some are less hydrophobic, but they are all mixed mode. So if the current version doesn't work for you, we can always try a new ligand structure for your purification.
Okay, I got it. Okay, excellent. So I have another question. Do you have any suggestions for separation of IgG Fab or FC with Nubia C prime resin? The answer is yes. I guess
doable.
It's doable. It will obviously depend. We think of these obviously as just another biomolecule that if we find a way to sort of tweak the selectivity, we can certainly do the purification. So I would say the short answer is yes, and I would focus also CHT. We've talked quite a bit about CHT today in the background. It still remains a very powerful sort of tool because of its chelation and cation exchange selectivity. So let's not discount that as a potential tool.
And you can also write to me. I can send you a slide to show you the separation of FC fragment, FAB fragment, and the full-length IgG on a CHT, so you can get an idea about the strength of the interaction and under what condition you will be able to elute the bound fragment from CHT.
Okay, excellent. Okay, I'll wait like one more minute. I think we have like 3 more minutes, but I think we got all the questions, unless someone is ferociously typing. But I hope well, thank you very much, and we really appreciate this kind of interaction. I hope we've demonstrated a level of expertise and mostly a level of openness that will allow you to sort of reach into our group and sort of discuss with us what's on your mind and really put our experts to work on your problems. And this is really the goal of this activity. Over and above the promotion of any given product or idea. It's really more of a sharing of the mind. So I'd just like to close with that and hope that you continue to expect great work from us. And I'd like to thank the presenter again, the organizers, the folks at On24, and remind you all to come back next time you see an invitation from us. And we'd love to hear from you if it was valuable. Thank you again for your time, and we really appreciate you coming to today's webinar. Thank you, and have a good afternoon and evening.